Hello everybody. Hi Martina, and today we present you the work I am currently doing as a postdoc in AstraZeneca. So, in the Department of Medicinal Chemistry, under the supervision of Christian, Jon, Leonardo, and Lars, I started to investigate on the ability of the current public available deep learning methods to predict the protein dynamic landscape. So in other words, I'm trying to understand if it is possible with these approaches to retrieve information on the internal dynamic of the protein. So actually, the first question that probably pop up in your mind, especially if you are not in the protein structural field, is why we need to investigate on the protein structure prediction with a computational approach. So are not the experimental procedure actually good enough to predict all the protein we want? And what are the benefits of an in-silico protein structure prediction, and how can improve such models? A lot of these questions have been already replied from the first talk. However, it was interesting for me when I started working on the protein folding field that when I was looking at the number of sequence deposit, for instance, in 2017, and I was comparing this number with the number of structures that have been deposited in the protein data bank, so 3D structure for the same year, actually this number was 3 orders of magnitude less than the number of sequence. So therefore comes the necessity to accelerate the protein structure prediction. But how? So, well, we can actually use computational methods like the one that I'm going to present you during this talk. Let's start to have a look a little bit more in detail about the background that led to the implementation of this computational algorithm. So the protein folding problem can be described as an intellectual problem at the intersection between biology, genetic, evolution, polymer physics, and statistical mechanics. So in other words, we want to understand how the 1D sequence dictates the 3D structure of a protein. Overall, the protein folding problem can be divided in 3 different problems. You have the folding code, the folding process, and the computational problem. The folding code aims to understand the thermodynamic forces that govern the interatomic interaction and turn the 1D sequence into a 3D structure. The folding process is instead more focused on the kinetic aspect of the protein folding. And last but not least, the computational problem that aims to develop algorithms for the 3D protein structure prediction. The dominant driving force, they can classify in 3 main groups. You have the hydrogen bond networks, so the interaction between donor and acceptor, that dictate the alpha helices versus the beta sheet. You have the van der Waals and the electrostatic long-range interaction due to the distribution of the charge for the sidechain, and the hydrophobic and backbone angle preferential that are actually correlated with the van der Waals electrostatic and the hydrogen bond network as well. Let's have a look a little bit more in details on the thermodynamic and kinetic aspect of the protein folding. So based on the Humphreys theorem of the thermodynamic hypothesis, the native structure of a protein is the one with the lowest Gibbs energy, and it's a function of amino acid sequence and solution condition. However, the folding problem is not only a thermodynamic problem, but also has a kinetic aspect. In 2000, on the Biochemistry Journal, Flux, Simon, and Baker observed that the folding speed of a protein is correlated with the percentage of relative contact order, which is a measure of the contact in the native structure. So higher is this percentage, faster will be the protein folding process. Furthermore, other parameters have also been correlated, like, for instance, the number of amino acids in helices, the total contact distance, or the secondary structure, just to name a few. However, if you're thinking about on how many possible conformations a protein must search in order to find its native conformation, it's actually an astronomical number. Indeed, based on the Levital paradox, if we consider to have a relatively small protein of roughly 100 amino acids, all the possible conformation based on the number of amino acids that this protein can have is roughly in the order of 3 to the power of 100 conformation. And if we think in that the protein can sample 10 to the power of 12 conformation per second, it will need 2 times 10 to the power of 28 years for the protein to find its native structure. But this is actually impossible, since experimentally it has been shown that the protein folds in a range of microseconds. Therefore, protein fold via pathways and not randomly. And this is extremely important from a mathematical point of view, because we can actually describe from a mathematical perspective the folding process. And indeed, from a mathematical point of view, you can describe the protein folding energy landscape as a funnel landscape with a lot of minimum at higher energy and only a few, and actually only one, with the lowest energy. So the funnel formulation of the energy landscape provides insights into protein conformational heterogeneity and protein chain entropy, providing a microscopic framework for folding kinetics. I would say that up to 3 years ago, the main two possible approach for predicting a 3D structure starting from a 1D sequence information were the knowledge-based approach. So where actually similar sequence is based on the assumption that similar sequence lead to similar structure, and usually has an order of similarity, we are around 30%. However, this approach is heavily based on the PDB structure, which up to date are around 80,000 structures. It seems a very huge number, but if you actually classify this structure based on the number of folds, you end up with only 1,200 different folds. And this is a very little number of pieces that we can use to assemble our folding. To overcome this problematic, another approach has been developed. It's called the de novo prediction, and it's for low to non-homologous sequence in the protein databank. This algorithm usually divides the sequence in fragments, and you try to find the fold for the fragment, and then put together the pieces and have the overall fold of the protein. So in the last 20 years, as it was presented in the first talk, there is a huge blind competition with the aim of shed light on the protein folding, and it's taking place every 2 years. Usually, this competition is extremely helpful for testing the state of the art of the protein folding algorithm methods. If we compare the first cast with the last one, with the Cas9, sorry, it was amazing to see that we improved the quality of the prediction with a factor of 2. And in the Cas9, it came out one of a very nice server for performing protein folding from Baker Lab, Rosetta. So in 2019, we observed actually assist to a revolution in the field. During Cas13, actually several groups showed that by applying a deep learning basic methods to the protein structure prediction problem, it's possible to generate a full level of accuracy for protein lacking homologous in the PDB. As it was explained this morning, the main data give as an input to these deep learning basic methods are coming from analysis of covariance from the multi-sequence alignment. So if you imagine to have your protein of interest and you perform a multi-sequence alignment over different species, you will see that, for instance, and you want to see how the red residue is covariating respect to, for instance, the green one, you will see that when the red varies as well the green, and vice versa. This means that these two residues that are far away in the sequence are somehow in contact in the 3D structure. And this allows a first big information that is coming from the distance prediction. So a pairwise matrix of yes or no contact. Lately, these predictions were taken one step further, and the covariance metric used to predict the probability distribution between two residues in a window between 2 and 20. This probability distribution is not only telling you yes or no contact, but is also telling you which is the probability that these two residues are a certain distance x. And you can use this probability distribution to convert in restraint and decrease the exploration of the conformational space. In the last CASP of last year, there was an outperformance from AlphaFold 2. Unfortunately, the code is not public available, as well as the procedure is not very well detailed. But the second best group, Baker group with the Rosetta software, actually both released the code and the pipeline, and is actually the core of our protocol for evaluating the possibility of looking at conformational variability. So the aim of our project is to propose a deep neural network protocol for investigating the variability of the ensemble, and not only the one that is the best model closer to the experimental one, but we also want to see if we were able to generate an ensemble of structure that resemble the experimental conformational variability of the target protein. So this is overall the overall pipeline that we are I currently use in AstraZeneca. So as I was telling you before, we have the core of the pipeline that is based on the Rosetta software from Baker Lab. In this first stage, we are not using template-based. We want to see how it was performing without any additional information, if not the one derived from the multi-sequence alignment. We couple the Rosetta with a multi-sequence alignment algorithm from the Zank Lab that provides a very deep MSA, so a lot of information that we actually need. This Rosetta coupled with the MSA provide 1,000 models that are rescored with the Hamiltonian. Also, Hamiltonian combine molecular mechanics term with bioinformatic local structure biasing term. We energy filter the model. We cluster based on the RMSD, and we pick all the centroids of each cluster and relax. At this stage, we are testing the log-pref-MD, so a molecular dynamic-based refinement that is only refine the sidechain. We are also planning in the next stage to use the Rosetta Deep ActNet for comparing in terms of refined methods. However, the current log-pref-MD pipeline that we are using in-house has to be modified since we didn't have access to the engine that originally has been used to develop the pipeline. So right now, the log-pref-MD work with NumD engine, and the data have been post-processing in PMD. When I started this project, I dig a little bit more in the literature, and of course, there were other possible algorithms that can be used in order to generate the initial histogram. We start with PROSPR, that is the public version available of AlphaFold 1, so the one from the CASP, of the CASP of 2019. The PROSPR pipeline provides up to the generation of the histogram, but there are no information about how to convert this histogram in potential restraint. However, as I was telling you before, TR Rosetta, so the transformed restraint Rosetta from Baker Lab, provide not only the code but all the pipeline. The main difference between PROS-PR and PR Rosetta is that PR Rosetta also provides the probability distribution for torsional and angle. And the code for converting the the code for converting the distance torsional angle in potential restraint is also provided. So for what concerns that the refinement protocol, we decide to move versus a MD-based code. And based on the availability of the public release, we decide to choose LogRefMD. And the results that I'm present in this talk are based on the LogRefMD refinement. So, but let's start to have a look at the real data. I will present here 4 test cases, and the reason why I choose this protein is because for the same sequence, we have experimentally 2 big different conformations. The first case is adenosine kinase. From the experimental point of view, we have 2 different conformations, the green and the purple, that depends on the presence or absence of a small molecule. So the challenge here is the dynamic rearrangement as a function of a small molecule. I run a short atomistic MD based on the X-ray structure. And when I plot the RMSD versus the time, I observed that actually there was an interchange in conformation between the open and the close. So I wanted to test the pipeline and see what it was the result by applying the pipeline that I showed you before. So I asked for 1,000 models, and here I'm reporting the plot of the Rosetta energy functions versus the RMSD computed for each of these models, a blue dot, versus the experimental 1AK. As you can see from the Rosetta energy functions versus the RMSD, there was a poor correlation. So I decided to rescore each of the model with another Hamiltonian. And I chose as an Hamiltonian the Awesome Hamiltonian. And by replotting the energy calculated for each model with the Awesome versus the RMSD, I actually noticed a better correlation. Just to give you a flavor of what does it mean in terms of energy, if I pick a model from, let's say, a badged energy, and I overlap the white model to the green one, I observed that there was a kind of very different conformation explored by this model with this energy. While if I pick a model from a good energy, from a lower energy, and I overlap with the experimental one in green, I observed that the overall fold was actually very close to the experimental one. Just to complete this analysis, let me show if I actually color code each of the dots based on the Rosetta energy function. As you can see, the Rosetta energy function, the good Rosetta energy, they are kind of spread all over the RMSD. So let me just give you this one. So I will give you this example and then I will finish. So by selecting and all the model based on the energy and filter it out with the mean plus 1 standard deviation, we run the algo methods and we look at the dynamic and we look at the cluster. So as you can already appreciate here, there is a kind of dynamic inside the cluster. We pick the first cluster and we start to refine. And I had looked at the overall backbone RMSD compared to the MD of the experimental one, as well as the difference in internal sidechain orientation. I compare our prediction with the TR Rosetta Solver prediction, and I observed that actually by rescoring an energy filter based on the Awesome energy, we were able to pick a nicer model. So we were asking for 2 conformational change, and we actually be able not only to retrieve the conformation, but also the intermediate state. So let me jump. These were other test case. I'm sorry for running out of time. So let me just go to the conclusion. And I'm more than happy to talk offline. So our preliminary result show this is the overall result. So I add other 3 test cases. In these 2, I was not able to get the 2 conformation, but rather an intermediate. And the difference between these 2 examples are mainly on the fact that the big conformational change here are due to the fold on unfolding part of the region of the protein. So in summary, the switch between a structural distinct state is crucial in proteins that are carrying out function as catalysis or molecular recognition. Although the current method have been tested for the prediction of static X-ray structure, it seems to be able, in principle, to explore the dynamic conformation space of the protein. And by combining deep neural network, molecular mechanics, and molecular dynamics, we actually observe a potential correlation between the residue-residue distance prediction and the protein flexibility.